Good morning, and thank you so much for the lovely introduction. It's great to see such a large crowd of people from a variety of different backgrounds. So thank you so much for the opportunity to present this work. So today I'll talk about some of the work that we do and give some case studies about trying to find linkages between soil health, plant health, animal health, and human health, based on the premise that all of these are probably linked together. So, as Lauren pointed out, our research really has two pillars. The first one is linking soil, plant, animal health to food nutrient density. So what we use in our lab are advanced mass spec technologies to profile foods. And I'll talk a little bit later about how we try to take foods beyond just protein and beyond just fat. Because unfortunately, we have dumbed down foods to usually single nutrients, but foods are very complex, containing thousands of metabolites that are capable of impacting our health. And then the second major pillar of our work is really also, and I don't know if you can see it, but we have a clinical facility where we perform randomized controlled clinical trials. So besides testing the food for nutrient density, we often feed these foods to people as well. And we look at how does that impact their health? And do we see this flow of nutrients from the plant to the animal and to the human, based on the idea that, of course, we as humans are also just animals. So I want to start with sort of painting the picture here, is that there's a half-truth to the saying, you are what you eat. If our grandmother tells us, well, Stephan, you are what you eat, it turns out she was about half right. But it points out that the way that we interact with our environment obviously shapes our health. And what we put in our mouth multiple times a day through diet, and this is really nice work from a group out of Israel that looked at a reference map of the potential determinants for the human serum metabolome. And I'll talk a little bit about what the metabolome is, but it's basically metabolites. And many of these metabolites within foods, right? Plants have metabolism, animals have metabolism. Many of these metabolites can serve as important nutrients to us. And then we also incorporate that into our metabolome within our blood. And what the authors found is that diet predicts about 50% of what circulates in our blood. So then the question is, and that's what we'll talk about today, specifically in regards to livestock. Am I what I ate? And are you what you eat? So we perform a lot of on-farm sample collection with farmers performing field work, and this involves collecting soil samples, oftentimes. This was work that we did with a gentleman, Alan Franz Lubers, a scientist at USDA ARS, and in this particular work, what I'll present about today, we wanted to compare grass-fed and grain-fed beef systems, essentially. What we had was we sampled the pastures on farms, and oftentimes we asked the grass-fed rancher, where's your nearest cornfield? And it turned out usually that was within a mile, so it wasn't hard to find. So we sampled soils on the cornfield. We sampled the soils of the biodiverse pasture. The cornfield, obviously part of it will go into livestock feed. A lot of it will go into ethanol fuel production, but it was a model of studying what happens when you grow feed for the animal, or what happens to the ecosystem if you just have it graze. So another thing that we did was we collected total mixed ration samples, pasture samples. As you can see, their makeup is wildly different, so you can kind of already see it, just like the soil. But in that case, we also collected meat samples, and I apologize for absolutely defiling these beautiful ribeyes. But in order to study them, we have to pulverize them on a liquid nitrogen and turn them into a very fine powder, and that way we can extract all the different nutrients from it for the different assays. What we do after that is we really try to study, besides just the primary nutrients, the protein, the vitamins, the minerals, we also try to study what is oftentimes called the dark matter of nutrition, right? Because we need to move beyond protein and fat and really consider the food matrix. What is nutrient density? Well, nutrient density, there's various definitions, but for me, it is more beneficial nutrients per unit product. And that can happen in a wide variety of nutrients. So even though if we look at a nutrition facts panel, and I don't know if you can see it here, but this would be a nutrition facts panel for steak. It gives you something about the protein, about the fat, cholesterol, and a handful of vitamins and minerals. And there's only 13 of those nutrients that appear on food labels, and you can't really see the difference in nutritional quality oftentimes from the current nutrition facts panels. Even within the USDA food composition databases, we track around 150 components in food, and they form the basis of our nutritional understanding, but it is estimated from food metabolomics work that food sources contain over 30,000 compounds that are capable of impacting our health, far beyond protein, vitamins, and minerals. And I certainly don't want to make the claim that we are able to study 30,000. I think we're still at the tip of the iceberg. We're able to maybe study 1,000 or so routinely. But what we're finding is that if you have a broader array of nutrients that you look at, you really start to see this difference between regenerative agriculture and conventional agriculture. Because why is that the case? Is, well, in a conventional system, what do we put back? We put back nitrogen. So there's no difference oftentimes in protein between conventional and regenerative agriculture. We also use mineral fertilization. So oftentimes those are not different either. We may formulate rations to be adequate in B vitamins, right? So it comes back to this idea that we are creating more simplified foods, and we are adding some nutrients back to the system, but we are not adding 30,000 nutrients to the system. And these 30,000 nutrients, they are often quite responsive to the way that we grow our foods or the way that we raise our livestock. So think of this also as complexity. So we study the food metabolome. Again, it's just a fancy way of saying that we study the metabolites that are found in foods, many of which are serve as nutrients to us. But then we study the human metabolome. And over time, I have come convinced that a healthy animal is healthy food, and probably a more nutrient-dense animal or nutrient-dense human is also a healthier human. This is not advocating for cannibalism, but that's basically how I would see it. And then the interaction with the nutrigenome, so gene-related expression, or the various of these metabolites, even though they may not have the classical energetic or caloric value, they impact our genes. They impact our cancer risk. They impact how our insulin sensitivity. They impact a cardiovascular system. So, and then we have a feedback loop there. So if we, for instance, look at the nutrients of meat, and today I'll talk, we in our lab, we study all foods. I always jokingly say, people, as long as you don't send a rock, I feel pretty comfortable about studying it. But today I'll talk about the nutrients in meat. And in the nutrients in meat, there's, again, estimated that there's more than 1,000 compounds in meat. These are various peptides, vitamin metabolites, energy metabolites, choline metabolites, omega-3 fatty acids, phytochemicals. And we'll talk about various of those in the presentation. So nutrient density testing, what is it really? Well, oftentimes we work together with farmers, also through our service lab, where people can submit samples. We process these samples in the lab. Oftentimes, especially if it's a food that there's not that much known about yet, we do what is called an on-target analysis. Basically, you look at anything that's under the sun to get a feel for what is in the food and how do you drill down on that further. And then oftentimes that's followed by targeted extractions where we extract fatty acids, run it on an instrument, extract the minerals and heavy metals, run it on a separate instrument, study phytochemicals, study B vitamins, study fat-soluble vitamins, etc. So all of these require different extractions to really accurately test the nutritional value of those. But it's all uses mass spec-based technology. So I want to start with really illustrating that grass-fed isn't grain-fed and grain-fed isn't grass-fed. When we studied 800 metabolites in this particular work of grass-fed versus grain-fed beef, we identified 825. And oftentimes what you hear as well, and people say this to me all the time as well, is that, well, there's maybe a little bit of difference in omega-3 fatty acids between grass-fed and grain-fed beef. But other than that, there's not much difference. But if you're only looking at protein and later on I'll also show you vitamins. That may be true. But if you're studying 800 metabolites, you can see that there's about a 50% difference. And I don't know if you remember from the earlier slide that about 50% of what circulates in our body is directly related to the foods that we eat. So you get a very much different phenotype. It is almost maybe one analogy to use. Let's say we split this room in half, and for a few months we will put half of you on a standard America diet and the other half on a Mediterranean diet. And if I were to ask you, well, after 3 months, do you think your blood will look different or your metabolome will look different? You'd probably say yes, absolutely. Well, the same thing is happening within the animal when you feed two very much different finishing diets, being grass-fed and grain-fed. But we will talk about the nuance later on, because grass-fed isn't grass-fed either, and grain-fed isn't grain-fed either. There's tremendous variation, and actually we found that some grain-fed samples might actually be more nutrient-dense than grass-fed samples, depending on the practice and feed that is being used. But we find the same thing in bison as well. So a 50% difference, so much larger than is perhaps appreciated at the moment. So we'll talk about not all 826 nutrients, because then we'll be talking until tomorrow, but I will at least describe starting with the fatty acids. And if we look at the fatty acids, forages tend to be very rich in omega-3s, and corn tends to, or total mixed rations tend to be not that rich in omega-3s. So end up with a different amount. This slide illustrates the omega-6 to 3 ratio. So the omega-6 to 3 ratio, typically a lower ratio in beef is considered beneficial, means you have more omega-3s, or proportionally to the omega-6, or at least you start to increase the amount of omega-3s in this case. So what is considered a good ratio? Well, typically we see that high quality grass-fed beef is about 2 to 1, 3 to 1 are typically very, very strong ratios. And you can see that here. And this was done as part of the Beef Nutrient Density Project, the work we did with the Bionutrient Institute, where we studied about 400 samples or so. And one thing that was interesting is that we clearly got the creme de la creme of grass-fed beef producers submitting the samples, because as you can see here, a lot of these fall in the average was 1.7, but a lot of these fall in about this range of having an omega-6 ratio of about 2. When we actually looked at these samples, all of these were sourced from a wholesale or grocery store. So this was just basically us walking into a grocery store and buying grass-fed beef. And I'm not going to stick my hand in the fire for it, but you have to really try to get your grass-fed beef up to a ratio of 10, because it essentially looks like feedlot beef. So, and I think that also is important about the transparency within our food supply. And a lot of this was, like I said, these were samples that we got from, and I think a lot of samples that we got from farmers that directly submitted, and these were, again, a lot of it also coming overseas or just walking into a grocery store and buying samples. At the same time, as I mentioned, grain-fed isn't grain-fed either. When we source from smaller family farm feedlots, which are also the people that submitted to this, we oftentimes see that they have actually ratios that are quite favorable at 5, typically less grain and shorter finishing phases. But we did find a very strong relationship between regenerative or rotational grazing on biodiverse pastures. The strongest relationship that we found was actually biodiversity. So plant diversity really increased the nutrient density of the beef. So one omega-3 fatty acid that doesn't get as much attention is typically DPA. We oftentimes hear about DHA and EPA, which are found in fish, but they're also found in pastured livestock products. But actually, beef is quite rich in DPA, which can be rapidly converted to EPA. And we see that these values actually are quite higher, and they're about 3 times higher in grass-fed beef than grain-fed beef. But again, notice this incredible variation with actually some grain-fed samples being higher than some grass-fed samples. Again, here, the strongest correlation, and probably the only correlation that we found, was with plant diversity. Of course, there's a tapering off effect. It's not that 200 plants is per se better than. I think the cutoff was about 8 or 9 plants, so quite that you have some diversity within your pasture. But what we found here in particular is that you typically getting enrichment in EPA and NDPA, which are important for cardiovascular health. And we actually, I'll conclude the presentation today also with some human nutrition work where we can actually see that this enriches the amount of omega-3s in the blood of people. Because one thing that you often hear is that, well, this is such a small amount, this has no biological effect. But this comes back to the idea of that if you maybe look at this on an Excel spreadsheet, you would say, well, this is not much. But it does seem like when you get this within a whole food matrix, that the bioavailability is probably higher. That is at least our hypothesis. Maybe because of the presence of phytochemicals that protect it from oxidation. You have other cofactors that can help with the uptake. This has not been studied in this context. What we do know is that if you take vitamin D from a pill versus from eggs, the bioavailability is about 5 to 10 times higher. So it could be that something like this is going on here as well, because despite the modest amount, you can raise the levels of the in the blood of people. One thing also that I want to point out is that the difference actually not in omega-6 fatty acids between grass-fed and grain-fed beef. It is actually quite similar. So it's really the absence of omega-3s in grain-fed beef that really drive that ratio up. And so by having fresh forages which are rich in the omega-3 ALA, you're really able to enrich the omega-3s within grass-fed beef. So cows are what they eat. If we have more omega-3s in the forage and feed, we have more omega-3s in the meat. And this is like a concept called the omega balance. One thing as you saw from the omega-6 to 3 ratio is that everyone hangs out at the bottom. So what the omega balance does, it basically scales it at a 0 to 100 score or percentage. So as you can see here, forages are very rich in omega-3s. About 80% of all polyunsaturated fatty acids found in forages are omega-3s. You see that this enriches the omega balance or the proportion of omega-3s in grass-fed beef, whereas total mixed ration samples are quite low. And you have grain-fed beef that is also quite low in omega-3s relative to total polyunsaturated fatty acids. Another study that we did was with the branches, and we really wanted to see also, well, what happens when you have more of these intermediate profiles as well. So we had a pasture-finished bison on a diverse native range, pasture-finished on limited forage diversity, but the forages were still very rich in omega-3s. Pasture-finished with free choice corn, which was actually, you see that you still have a very favorable omega-6 to 3 ratio. And these were pen finished with free choice access to hay and corn with low stocking density and high stocking density. You get a more intermediate profile. And this was from a more traditional feedlot where you get an omega-6 to 3 ratio of 8, which is about typically in line with what you see in feedlot beef and bison as well. The reason why we're particularly interested in is that in perhaps traditional or mixed farming systems, it was oftentimes quite normal to maybe run your animals over a spent crop field, and maybe you would consume some grains or would consume some byproducts. So in this work, we're particularly interested in also what if we feed some of those byproducts to animals. And with the bison, it was particularly because they lose a lot of weight over the winter. So if they can maintain some weight by maybe eating some grain, that could be potentially a beneficial thing, because I do think that one important way of looking at it is that it's not really black and white. It is gray. For instance, we also worked with a farmer that was able to get some spent brewer's grains from a local brewery and fed that to the animals on pasture. Well, they were indistinguishable from grass-fed. We could not. It's that he told us, but we also use predictive modeling where we're trying to work on, can we identify, based on all these metabolites, all these nutrients that we mentioned, can we identify grass-fed beef? Also, are there tools for authentication that can be used? Because clearly some of those grass-fed beef samples, well, like I said, I'm not going to stick my hand in the fire for it, but I'll keep it pretty close, because those are not, those don't look like grass-fed beef samples, right? So we have to be, if we can authenticate that, I think that will lift the entire industry. But some of those samples that were pasture finished with small amount of grain, they registered as grass-fed beef samples. They were not distinct from that. So another way of looking at this is really also pasture finished, and maybe that's an important nuance that we need to explore in the future. This is from other work that we did. Also, a lot of farmers that submitted samples to our lab, to our service lab, where we study nutrient density for them and give them their data back. As you can see here, there are similar findings made in other species as well, where corn, soy for free pork with daily pasture, omega-6 3 ratio 5. When they were fed 50% corn, soy, about 10. And this is really just a corn, soy, fat pork with no pasture. It goes up to 30, which is incredibly high, as you can see, because our current standard Western diet, we consider that to be very high, in omega-6 3 ratio of 20. But it goes even far beyond that. So in monogastrics, there's actually very strong opportunity to improve this as well. And probably the effects of feeding are much more pronounced. And we also have wild deer that registers about an omega-6 to 3 ratio of 2. So one of the questions you often get is, grass-fed beef a good source of omega-3 fatty acids? Well, there's no US guidelines, but there are European guidelines. And European guidelines would consider anything above 40 milligrams per 100 gram, as you can put it on front of package labeling, sources of long-chain omega-3 fatty acids. And perhaps in the future, we could do something like that in the US as well. But as you can see, there's a good amount of ribeyes from that study that reached that type of level, and technically in Europe could put a package claim on being a good source of long-chain omega-3 fatty acids. So we also looked at vitamins within that study. Again, the main thing that we find is that there's this incredible variation, but there were no difference in B vitamins. If anything, some of them, like vitamin B5, which enriches grains, was a little bit higher in grain-fed beef. And this is also partly by design, of course, right? Total mixed rations are designed to provide adequate protein, adequate energy, adequate B vitamins, adequate minerals. So it's really in the omega-3 fatty acids, in the phytochemicals, which I'll talk about later, as well as in fat-soluble nutrients, like the carotenoids, like the tocopherols, that are being turned into vitamin A, into vitamin E, into vitamin K, where grass-fed beef really distinguishes from grain-fed beef. We also see in this broader array of peptide, or what's sometimes called bioactives, the dark matter of nutrition. If you study a large variety of nutrients, you see that there's this 50% difference that starts to appear. But it's not really in protein and vitamins and minerals. And this is also oftentimes what various systematic reviews would suggest, because people will tell me as well, various studies find that there's no difference between organic and conventional, but it very much depends on the nutrients that you're looking at. If you're looking at protein and vitamins, typically no major differences, but that's also because those are being provided or inputted within the system, of course. So again, we find this incredible variation here, but on a group level, we didn't find significance in B2 and B3, with B1 being a little bit higher, because thiamine is rich in grains. So you get that a little bit being a little bit higher in grain-fed beef. Pantothenic acid, vitamin B5, was quite a bit higher in grain-fed beef, about 1.5 times higher. Reason being is that vitamin B5 is rich in grains. So we did find large variation amongst individual, but no clear separation between grass-fed and grain-fed. And I told you this is partly also by design, because you want those nutrients to not be different. Why? Because they're very important for growth. So that's why rations are often formulated to meet those needs. Now, fat-soluble vitamins, we do find differences. Fat-soluble vitamins, similar to in our food supply, right? What do we fortify our ultra-processed foods with B vitamins, with minerals, but not with fat-soluble nutrients because they don't have a very long shelf life. Maybe the exception would be milk because we drink that very routinely. So again, in the fat-soluble nutrients, which are very rich in green forages, carotenoids, the tocopherols are very rich in green forages. They start to become enriched, as you can see here. And then retinol, the vitamin A as well as vitamin E, as well as vitamin K2, which was significantly elevated in grass-fed beef. So cows are what they eat. Well, if we look at the example here of vitamin E, about 1.8 times higher in grass-fed beef versus grain-fed beef. Then when we studied the forages versus the TMR samples that we collected, we can see that the forages are quite rich in alpha tocopherol, about 20 times higher, whereas the total mixed ration is really low in vitamin E. So this is probably the contribution from the pasture phase, with little contribution from TMR. So you see this decrease, and all right, there we go. The clicker is clearly slower than my brain works, so I guess that's good for me. But the phytonutrients, they transfer from plant to animal. So really another thing that we really wanted to look at is that, well, we know that forages are rich in what is called phytonutrients. It's a blanket term that really refers to the other 30,000 metabolites that are particularly within plants, and we're interested in studying how do they get biotransformed within the animal, and what are the effects on animal health and then also our health. So plants they produce these phytochemicals. They cannot run away, so they have to find another mechanism to protect themselves. But when we or cows eat plants, and luckily we eat different plants, and that's actually a very good thing, is that we can upcycle unique compounds from forages through the animal, and that are again getting biotransformed and can make its way into us. Oftentimes we're able to access medicinal plants that we would otherwise not be able to access, and I think that's a very important nuance. So cows are what they eat. Phytochemical richness of forages. We studied various forage samples within the study. We find here that this is much higher than corn-based TMR, and we studied corn only. It barely registers. So corn is a good source of protein and energy, but provides little antioxidants to the animals. So this was something that we really, I won't go into the details too much, although there's maybe an interesting sidebar, is that once we started this, people had studied this in the past using more UV vis, so photospectrometry. So we were studying, and there's some reports in the literature, especially with the French and Italian scientists, that maybe compounds like benzoate, which is very rich in green plants, would also appear in the animal source foods. Then we started to study this on a mass spec where we can actually measure the masses, and we're like, well, it's not there. So then we had to also figure out, well, and luckily we learned a lot actually from human breast milk studies, is that, because we're also just mammals, right, is how do these things get converted into downstream metabolites? And for instance, hyporic acid is one that we really found to be a good probably biomarker of grass fed. It's a major antioxidant. It's associated with reduced odds of metabolic syndrome in humans. In the absence of chronic kidney disease, if you have chronic kidney disease, this will also be sky high. So nuance or context matters. If you eat a bunch of blueberries, it'll also go up. But it's a biomarker of phenolic intake in mammals, in cows and humans, as this compound is produced from phenols within the gut microbiome. And we see that this is, in some cases, very, very high within grass-fed samples. Now there won't be a quiz on this at the end, but this is basically to illustrate that if we have pasture versus TMR, and this is the fold higher, is that these phytochemicals, they are 100 times higher, 889 times higher, 79 times higher, 50 times higher, etc. You get the gist of it. They're much higher, these phytochemical antioxidants. On average, 276 times higher in grass-fed, or in grass versus TMR. You do get a little bit of a dilution effect, but it's still 6x higher in grass-fed beef for various of these compounds. So we see that those compounds become particularly enriched within the animal, and actually might provide more complexity, more flavor. Another thing that I think I talk a lot about nutrient density, but actually the way that we first got into this work was by looking at, and the French and Italian scientists, they were not really interested in the health benefits. They were just thinking, well, if we graze our animals on the alpine pastures or on the Sardinian pastures, how can we make our cheese look better so that we can sell it to maybe Americans? No, I don't know, but that's really why they probably wanted it for themselves, actually. So that's one important thing to note. But then, as a nutrition scientist, I was looking at it, I was like, wait a minute, these are also have potent health benefits. So that's how we really got to study that. Another thing I want to talk about is terroir. Terroir, it means the territory. It means that if you eat beef from Kansas or from North Carolina or from Texas or Oklahoma, it will taste different, because you're tasting the terroir, you're tasting the territory, you're tasting land, right? So what we see is these very unique compounds appear in some samples that are not found in any other samples as well. And this is particularly a anthocyanidin, actually, that is probably found, that we know is found in purple and blue plants, and it actually makes its way into the animal a little bit as well. S-equal, which is an isoflavone studied for anti-cancer properties. Now, about only 30 to 50% of people can produce it, but the animal actually does a pretty decent job at producing it, and then it's another way of getting some of those compounds in. Ethyl gallate, another example. Again, I won't go through them all, but apine, also natural flavonoid. Again, you have these very unique compounds that you can find within grass-fed beef. And in this particular assay, we studied about 305 phytochemicals. Since then, we have raised it to about 500. This work was done probably maybe 9 months or so ago. But we find a wide variety of them. About 205 were detected in grass fat, 187 in grain fat. But this was driven primarily by the producers that were, I mean, I don't know if it's not a proper scientific term, but I call them an artisanal feedlot. Basically, it's maybe a smaller family-style feedlot where the farmer or rancher may finish their animals for 30 days or 60 days on some corn and hay to get some extra marbling. And those samples, while they technically would be labeled as grain fat, as I mentioned, they are actually still looking very nutrient dense. Now, history always repeats itself, right? This is an anecdote that I want to highlight from Warren Angus Ferris, who was an explorer who traveled the Great Plains from 1830 to 1835. And he mentioned that during those adventures, Ferris grew fat on bison, as indigenous people had done for ages. And he notes that bison in poor flesh were on the worst diet imaginable. I'm picturing an overgrazed monoculture. But as the animal fed on diverse mixes of plants, no other meat could compare. With it, we require no seasoning. We boil, roast, or fry it as we please, and live upon it solely. What seems to be most singular, we never tired or disrelish of it, which would be the case with almost any other meat. I'm pretty sure that he didn't know what he was referring. Well, he did know, but I'm pretty sure he's referring to the phytochemicals that are found within that meat, basically following indigenous practices that would be able to taste that meat, taste that terroir, taste those phytochemicals, the flow of nutrients from forage within the animal. And I think that's what we're seeing on the mass spec work. So the flavor of meat and milk is influenced by the phytochemical richness. They have potential health promoting effects. So flavor and health are related. Arguably, the best mass spec is the one that's on your tongue, because if you have food that is more nutrient dense, you taste that, you register that immediately. Then if we run it through a mass spec, we might find, well, this blueberry, or as Herb will talk about, this citrus fruit, this orange tastes a lot better, has a lot more flavor complexity. If you then run it on a mass spec, then a compound like naringenin might be X amount higher, right? So these are things that we can definitely taste as humans as well. So animal health, human nutrition, are they linked? Well, one of the questions is, and there's ongoing research on whether a healthier animal results in a healthier human. I don't have the definitive answer for you, per se, today. what I do feel pretty comfortable about stating is that it's definitely making the animal healthier. If you look at something like homocysteine, which we also use as a biomarker in humans, or 4-HNE, which is an advanced glycation end product. And this is another interesting thing that to notice is that, for instance, 4-HNE, red meat is sometimes considered carcinogenic. Now, granted, if you're from California, everything's carcinogenic, but you know, in general, if we can lower that amount by the way that we raise our animals, that could be a benefit. And we see it here, less oxidative stress in the animal. So the animal's healthier, then what does that do to our health? So we see here that the animal shows a more healthy metabolic phenotype. And my background is actually in exercise science, and we were taking biopsies from humans, athletes, but also maybe people with chronic metabolic disease. When we first started to look at the metabolomics data from the animal, the parallels were very interesting, where the grass-fed animals look more like athletes, whereas the grain-fed animals, I'm not saying they have metabolic disease, but they start to show a phenotype that is very similar to the human phenotype, where you have impaired glucose metabolism, the mitochondria is not that healthy, you have more oxidative stress. With the question is, what does that do to us? Well, but I want to tie it back. What are the relationship with soil health? This is where we started with, right? The monoculture cornfield, the biodiverse pasture. When I showed this to my wife, she said, well, the picture says it all. And I said, and she's also a scientist, and I said, I agree, but we also need to actually study it and study the data, of course, right? We cannot rely on this, but it has been really interesting when we do this work with farmers. Is a lot of times I feel like we're verifying what farmers already know. So we look at soil organic matter, total exchange capacity, various macro and micro minerals. So we're generally enriched in the pasture soil samples compared to the adjacent cornfield. So overall soil health seems to be improved on a group level. We did this work together with Alan Franz Lubers. And what we concluded from this paper is that the study provides evidence that rotationally grazed perennial grasslands can store more soil organic carbon and nitrogen and improve soil surface stability conditions compared with neighboring croplands producing commodity crop-free grains for feedlot finishing. So this paper was published 2 years ago, where we came to that conclusion that managing the field as a pasture versus growing commodity field crops improves markers of soil health. Are there also biomarkers that we can study within the metabolomics assay? And the answer is yes. I don't know if you ever heard about the compound ergothioneine, but ergothioneine is produced by relatively few organisms, mainly within cyanobacteria and fungi within the rhizosphere. It's the only known source, and it makes its way up through the food web into the plant. And then, as we can see here, also higher levels within the grass-fed animals. Histidine beta is its metabolite, its cousin, and it also gets enriched. So, the final part of this talk: is there an impact on human health? Animal health seems to be improved. Nutrient density seems to be improved. Can we then also improve the health of the consumer? And when we link animal and human health. So this is fresh off the press, because we completed this study in October 2025. Initially, we did a shorter trial. Then we followed it up with a longer trial as well. This was 4 months. The original one was 6 weeks. When we completed the study, we had a randomized controlled trial where we recruited people. They either were consuming they get randomly assigned to the conventionally raised meat, and this was beef, pork, chicken, and eggs, or the pasture raised group. We gave them all of their meat and eggs for 16 weeks. They would come to the lab multiple times. We collected urine, stool, questionnaire data. Then we looked at a variety of outcomes. We have the RBC data, which I'll present to you today, the metabolomics data. We are going to analyze that probably this pretty soon here, November. But it takes a little bit to extract, because it's a lot of samples that we collected. But this is one of the longest and largest studies comparing pasture finished versus conventional. So the average omega-6 to 3 ratio in whole blood of Americans is about 9. Our dietary intake is about 15, but the average is about 9. We were interested, well, if we're feeding pastured animal source foods, can we then improve that? Well, the folks within our study, they didn't have quite a ratio of 9, but at about 6. So this is at the end of the trial. The conventionally raised group that ate grain-fed beef and then conventional pork, chicken, and eggs has an RBC omega-6 to 3 ratio of about 6. We were able to bring that all the way down to 2 by eating pasture-raised animal source foods. Mind you, we didn't feed them any fish or anything, of course, because that would be a confounder. But it does tell you that pasture-raised animal source foods can move the needle on omega-3s. Now, I know this is not a hard outcome, but if you do so for 30 years, in theory, you should lower your risk of a variety of diseases, Alzheimer's, heart disease, cancer, etc. So, again, here, this was a very, very interesting finding, and one of the first studies in North America. Historically, there have been studies in Australia and the UK that had studied this. But yes, we find that pasture-raised animal source foods can meaningfully move the needle. And these are some of the initial ones. And again, DPA being very rich, EPA, DHA, and DHA. DPA, which kind of sits in the middle, also in the metabolic pathway, being very much enriched within the pasture-raised group. So we will study a broader metabolome profile, trying to answer those questions. Does having more 4-H&E, more oxidative stress markers in the animal, how does that impact our health? How does the flow of phytonutrients from forages that are transformed into these mammalian antioxidants, how do they impact our health or our metabolome? Those will be our ongoing investigation that we're working on, because especially randomized controlled trials, they take time. What I do note or conclude, what we do conclude from this work, is that phytonutrients become concentrated in the meat and milk of animals finished on biodiverse pastures. They give more complex flavor to it. They improve the health of the animal. And some of them have known human health effects, but we just don't know if from the consumption of animal source foods they can contribute to that. We're finding increased levels of omega-3 fatty acids in grass-fed beef, with little difference in B vitamins, but higher levels of fat-soluble vitamins. We also find that omega-3 levels can be raised in the blood of people. But I do also want to bring it back to sort of the bigger picture. We have done another study too, where we, and in the interest of time, I didn't present on this here, but we fed people all foods from regenerative agriculture versus conventional agriculture, but they were consuming a standard American diet. If you eat whole foods or eat these things as part of a healthy diet, that is key, right? If you, I always jokingly say, if we turn a, if you were swapping out pasture-raised meat, but eating a food or a diet very rich in ultra-processed foods, you're not likely going to move the needle as much as you can. So bigger picture, diet quality is a main consideration. Eating pastured meat as part of a standard American diet is also not going to transform your health, but that's part of a healthy diet, rich in whole foods, that I know taste is subjective, but that at least have more flavor complexity. It may be an important piece of the puzzle. And we have a paper, a systematic review that's about to come out, where we actually looked at all the literature that was out there and actually found that there's quite a variety of compounds that are not found in the human metabolome or in the human diet, but that are being upcycled from the animal. And now arguably you could say, well, they're found then in the human diet, but that make their way through the animal, because the animal really consumes plants that you and I cannot consume, providing another avenue by which we enrich the phytochemical richness of our diets, while providing omega-3 fatty acids and important critical fat-soluble nutrients. Now, I am, in many cases, also just the messenger. We had many people that worked with this with us on this together. Started this work while I was back at Duke. Also various collaborators at USDA ARS, Scott Kronberg, David Archer, as well as the work with the Bionutrient Institute, the Beef Nutrient Density Project that I presented on today as well, and the team at Utah State that works with me. And last but not least, a lot of this work has been made possible from farmers that submit samples to our lab and have them tested. So, and also provide the resources to test the sample. So we're incredibly grateful for that as well, because the beef nutrient density project, while we had some USDA funds for that, that was essentially, a lot of it was crowdsourced and by farmers and foundations. So really grateful for that. And with that, I'd like to conclude this talk, and thank you so much.
We have time for one question. Is there anybody who has a question? Who wants to be the one? Yeah, we are live streaming. Someone asked you to speak into the mic.
Do you think that the phytonutrients from the plants are more bioavailable when we eat them via the meat versus eating the plants themselves?
It's certainly a plausible hypothesis, so I'm glad you pointed that out. We know that heme iron, or iron coming within the heme structure from animal source foods, is more bioavailable. Also, we do know that some of these compounds, they have to be transformed by our liver, but if we already get them pre-processed, that could be that to it. Yes, that is a working hypothesis, but I don't have the hard data yet to either refute or confirm that. But I agree. So you're onto something.